Hello Biology 230 at Tuskegee University. This is Dr. Griffin again, and we're going through Module 5, where we are focusing on cellular respiration. Now, the first major step of cellular respiration that we'll discuss for this module is known as glycolysis. And glycolysis literally means sugar splitting. And so think, we are splitting our sugar. Well, what's the sugar? Well, the sugar in this case is glucose, a monosaccharide. That's reviewing some of your earlier work that you've done in this class. During glycolysis, glucose is oxidized to the molecule known as pyruvate, and all this occurs in the cytoplasm. Per one molecule of glucose, 2 molecules of ATP are consumed during glucose. And per one molecule of glucose, 4 ATP are formed, 2 molecules of the reduced form of the electron carrier NADH are created. So 2 pyruvates are formed, as well as 2 molecules of water are formed. And if you look at what it was invested and what was created, we net during glycolysis 2 molecules of pyruvate, 2 molecules of water, 2 molecules of ATP, and 2 molecules of the electron carrier NADH, as well as 2 protons are created. And all this is very nicely diagrammed in figure 9.8. I'm not going to go through all the diagrams and the steps during this lecture, but I want to point out the take-home message is that during glycolysis, which occurs in the cytoplasm of our cells, it's oxidizing glucose to 2 pyruvates, and this is going to generate a net of 2 ATPs. So we form 2 new ATPs by splitting sugar, and we literally are splitting sugar in half, and you can see this very nicely again in figures 9.8 and 9.9 in your Campbell textbook. Key enzymes and key steps that you should be very familiar with for this course are hexokinase. Let's think of what that word means. Well, hexo means 6, and as you remember, kinase is an enzyme that phosphorylates. So therefore, hexokinase is probably an enzyme that adds a phosphate group to a 6-carbon sugar, thus glucose. And there we have it. This is actually the enzyme that uses the first molecule of ATP, phosphorylating glucose. Again, that is hexokinase. The next key enzyme is phosphofructokinase. Again, let's look at the name. Phospho, we know there's a phosphate group involved. Kinase, that means it's probably going to be adding a phosphate group. And fructo, well, that fructo is the fructose, the type of sugar that is being phosphorylated. And in fact, phosphofructokinase uses the second molecule of ATP in glycolysis. Specifically, it phosphorylates fructose 6-phosphate. The name pretty much tells you what it does, okay? Triose phosphate dehydrogenase. This is the third key enzyme and step that you want to be very familiar with for this course. That is where we're going to produce NADH during glycolysis. Phosphoglycerokinase. This is produces the first set of ATP. This is key. This is where the first molecule of ATP is made. This is part of our energy payoff phase of glycolysis. And last, but certainly not least, is pyruvate kinase. Again, we're getting some phosphorylation, and this kinase, pyruvate kinase, produces the second set of ATP molecules, and it is the very last step of glycolysis, and thus produces the end product of glycolysis starting from glucose. Then this end product is known as pyruvate. And this is all very well nicely diagrammed in 9.9, and we have a number of exercises available to help you understand how these reactions take place and the importance of them, of breaking down glucose, splitting it into 2, and producing these 2 pyruvates. Just some takeaway keynotes again. Each molecule of glucose will yield 2 molecules of pyruvate. ATP is produced in glycolysis. Remember, we're still in the cytoplasm. All this takes place in the cytoplasm and produces ATP in glycolysis via substrate level phosphorylation. If you're not familiar with that, go back and read the first part of chapter 9. There are two main phases of glycolysis. The first being energy investment, where we are using ATP to phosphorylate the sugars, and then the second phase, the energy expenditure, where we're using kinases to phosphorylate ADP to create ATP and produce key products, the main one being the last product, pyruvate. Note that glycolysis is not dependent on the presence of oxygen. Look through all of those equations and those reactions that are associated with glycolysis. Not one requires oxygen as a reactant. Therefore, glycolysis can take place in the absence of oxygen. Now, if oxygen is present, pyruvate can then be oxidized and the citric acid cycle can and will take place. And we'll talk about that in our next video for Module 4. Module 5, sorry. Well, this is the end of video 3 out of 4 for Module 5. And don't forget to view and learn from the next video. I look forward to seeing you soon, Biology 230. This is Dr. Griffin signing off for Tuskegee University. Have a great day.